Hello. This time we will talk about what happens to those amino acids and peptide after protein is digested. Right? So it will be related to amino acid absorption or peptide absorption and what happens to them once they are absorbed, what happens in the intestine. Right? Let me share my screen. Okay. Right. Next few seconds. Okay, I think you can see this screen with a white background again. Okay. So let's talk about the absorption of amino acids and the peptides after protein is digested. So they are coming, we are talking about this, the end product from protein digestion. Think about what we learned last time. Okay, and then also, what happens to those amino acids in the enterocyte or intestinal epithelial cells after being absorbed? So we call that first pass metabolism. Okay? Those amino acids. Let's talk about amino acid first. When the proteins are digested to individual or free amino acids, they are absorbed into the enterocyte, which is the intestinal epithelial cells in the small intestine. Okay? Not much in the duodenum, but mainly in the jejunum, the case of mammals, or jejunum and ileum in the case of avian species. Um, there are 20 different kind of amino acids provided from the protein digestion. So we're talking about 20 different stuff coming into the cell. Okay? And there are transporters responsible for the absorption of amino acids. But remember, those amino acids, they have all different characteristics depending on the R group. Some of them are hydrophobic, some of them are hydrophilic, and some of them are basic, some of them are acidic, aliphatic, aromatic. So the characteristics of amino acids are all different depending on the R group. So their absorption through the transporters will be also related to the characteristics of amino acid. So transporters, this is not only one transporter, there are many different types of transporters responsible for to handle the absorption of all these 20 different amino acids. So depending on amino acid and depending on what transporters are involved, it can be active transport or passive transport. When we say active transport, it means when amino acid is absorbed, it will spend energy. Passive transport means it doesn't require energy to be absorbed. Okay? And here, I'd like to introduce a term called system. And system is the collective term that we use for the amino acid transporters. Okay? We call them system. Okay? System, this is not only one, there are many of them, and they are functionally different related to what amino acid will be uh, taken by what system. The goal is for the enterocyte, the intestinal epithelial cell, to absorb the amino acid from the luminal side and pass that to the systemic circulation or blood circulation. So if you see a nicely drawn one enterocyte or an intestinal epithelial cell, at the top here, which is the apical side, top. You see a very finger-like structure. How do you call this? I'm sure you learned. It's a microvilli. It's not a villi, it's a microvilli found on the apical membrane of enterocyte. So this is the lumen, the luminal side or apical side here. The other side is a basolateral lateral membrane, and uh, this is the inside of the body. Okay? So there are systems on apical membrane. Also, there are systems on basolateral membranes too. Okay? So from the lumen, the amino acid will come through the apical membrane, through the systems, and then will exit the enterocyte through the systems on the basolateral membrane as well. Okay? So there should be uh, they will use the transporters at two times, at the apical membrane and basolateral membrane. Let's talk a little more about the system. They are functionally identified, and each system are responsible for transporting different types of amino acids. Okay, here, when I mentioned about system, I said there are many of them, so they have a different names. So how do we name the system? Depending on what amino acids are transported through this protein. The system is protein, okay? The membrane protein. Okay? System is named based on their preference of the amino acid preference. As examples, there is a system ASC, and there's a system GLY, and there's system L, right? So when you see the system ASC, okay, this is the system amino acid transporter responsible for three amino acids, alanine, serine, and cysteine. So this particular system is called system ASC. The case of system GLY, it means it's glycine. So we can see that this system will be responsible for glycine absorption. And the system L, here L stands for large neutral amino acids. So the R group of these amino acids are large and bulky and neutral. So those are leucine, phenylalanine, and tryptophan as an examples. Okay? So this system L will be responsible for absorbing, transporting the leucine, phenylalanine, tryptophan. So that's how we can find out what amino acid will be transported by which system. Okay? And often this name system, would have additional letter like X, Y, Z, or minus, positive, plus, or zero. Okay? When you see this X letter together with the name of system, typically those are the system responsible for the ionic anionic amino acids. If you see the Y, and those are system responsible for cationic amino acids, and the Z is neutral. Same for negative, minus anionic, and positive is cationic, and zero is neutral. So it is a little complicated because there are so many systems, but some of these examples may help you to find out, okay, what amino acid will be absorbed, transported by this particular system. And there are many different systems, but they have some overlapping specificity. One particular amino acid can be transported by multiple systems. Okay? And one system can transport several amino acids. So by doing that, I think we can maximize the efficiency. Right? And later, I'll show you a big table showing the different types of systems. Okay, but before that, I'd like to introduce different kind of nomenclature, the different types of naming of the amino acid transporter, which is called solid carrier. So this is a group of membrane protein, but this some of them are responsible for absorbing the amino acid. So the solid carrier, some of them would have a function of carrying amino acid or transporting the amino acids. So solid carrier is in short, written as SLC, SLC. So if you see SLC, that means solid carrier, and those are the proteins possibly having a function to absorb or transport amino acid. So this is different way of naming the amino acid transporter. As an example, SLC1, okay, SLC1, this protein number 1, is an amino acid transporter, especially responsible for the glutamate and other neutral amino acids. Another example I can show you is SLC number 38, this protein number 38. This is a sodium-dependent spending energy carrying neutral amino acids. Okay? In this case, SLC38, this amino acid transporter, is actually same as the system A, when with using different ways of naming the amino acid transporter. So you will see different ways of expressing naming the amino acid transporters, okay? System or SLC. Okay? Well, so it is a little complicated. It's confusing, actually, right? So I'm showing you the names. There are very different, many different amino acid transporters. Okay? I'm showing you two different ways. As one example here, if you see LAT1, that's the system, name of system, system LAT1. But this amino acid transporter is same as solid carrier number 7A5, SLC 7A5. This is same thing, but with different names. Another example here, let's see here, the system GLYT1, that's same as SLC number 6A9. That's the same protein, same amino acid transporter, okay, responsible for the same amino acids. Okay? Do I need to know all of this? No, we have this table. If you need to know, we can look at the references. So, but here the goal is, the objective is to show you, okay, there are so many different kind of amino acid transporters responsible for different kind of amino acids. And one amino acid can be carried or absorbed through several different types of transporters as well at the same time. Okay? But depending on the transporters, there are symporters or antiporters or uniporter. Symporter means when amino acid is moved from the outside or one side to the another side of membrane, the cation is moved together. Often this is sodium or hydrogen, okay? And amino acid and cation, they're moved at the same time. Antiporter means when amino acid move from one to the other side, and another amino acid will move the opposite side. So it's kind of exchanging. Okay? Uniporter is as one way. Okay, the amino acid will be moved from one side to the another side, or another side to the previous side. Is the uniporter. Okay? The symporter most likely is sodium dependent or hydrogen dependent active transport. Antiporter also mostly is a energy spending active transporter. But the uniporter will be most likely is facilitated is diffusion or passive transport, not spending energy. Okay? All right, and if I show some of the overall how this work is, we already learned how protein is digested to single amino acid. There are many proteases working here. Also, protein can be partially digested and becomes oligopeptides, and or can be a little better digested to dipeptide and tripeptides or tripeptides. If we talk about the amino acids will be absorbed going through the apical membrane of enterocyte. Okay? This will be done by so many different kind of systems or SLC. Okay? They could be sodium-dependent transporters or not, or so they are either transported spending energy or not. So it can be either active transport or passive transport, depending on the systems and depending on the amino acids. On the other side, basolateral membrane, here there will be another sets of transporters waiting for this amino acid to move from the cytosol of the enterocyte to the systemic circulation, and this can be antiporter, mainly is antiporter, whereas the apical membrane, the main is the symporter. So that is a little different thing, little difference between apical membrane and basolateral membrane in terms of amino acid transport. But both of them either use energy or not. Okay? In the case of dipeptides and tripeptides, they're already small enough and being efficiently absorbed into the enterocyte by different types of transporter. It's not a system. It's not SLC. It is called peptide transporter, PPT, type 1 or 2. In the case of type 1, PEP1 found in the apical membrane of the enterocyte, and it is a symporter, and it will use hydrogen. So it's a hydrogen-dependent energy spending active transport. Okay? And it enters the cell, the cytoplasm of the enterocyte. The inside, that can be most likely will be further digested by dipeptidase into single amino acid and then moved into the blood circulation by the amino acid transporters and the vasolateral membrane. But some of them can still be transported to the circulation through the peptide transporters as well. In the case of oligopeptides, okay, longer than tripeptides, they still need further digestion by the brush border enzymes called aminopeptidase. We briefly talked about this in the previous lecture, and this aminopeptidase will further break down oligopeptides into dipeptide, tripeptide, or single amino acid, and the end product will be absorbed. Okay? So this is the kind of overview of how it is organized here. Amino acids, dipeptide, and tripeptides will be absorbed. Okay? And those are energy spending or not. And there are many different types of systems or SLC are involved. Okay? If I summarize here a little bit, peptides here. Peptides mean dipeptides and tripeptides, and AA means amino acid, free amino acid or single amino acids. In the case of peptide, dipeptide, and tripeptide, their apical uptake is not uptake. Apical uptake. It is symporters, and they use hydrogen. It is not 100% is a mainly use the symporters, and they use mainly use hydrogen, so use the energy spending process. For the bilateral transport, it will be mainly the uniporter on one side, so it is not using hydrogen, so it's not spending energy. In the case of amino acid, for the apical uptake, it's mainly symporters, but some case of antipoders or uniporters as well. In the case of symporters, most likely it will use the sodium. So it is energy spending is active transport. For the basilateral transport, mainly is antipoders and uniporters, right? So they may use energy or not, right? So that's how we can summarize the absorption of amino acids and peptide. I know it's a little complicated here because there's so many transporters here. Um, if you find the papers, either research paper or review paper from 2000 or 2025, you see that there are changes all the time. There are more are found and updated. So it'll be good to check some little older paper and the newer paper and compare how they changed. Okay? Actually, I put the two references so that you can read on the Moodle website. So you can see one paper from 2000, another one from 2000, like 20. Okay? So you see how they are changed as well. Okay, this is actually I have a copy of part of the tables here. This is one. It's from the 2019. Well, there are so many of them, and this table they organized this table. The result in this table showing different systems and matching SLC and what amino acids are used by these different systems or SLC. Um, Okay. We talked about this already here, so I will just move quickly of this page. Okay, I'll move it. Now, we so far we talked about what happens in the small intestine. Okay, so that the jejunum in the case of mammals and jejunum and ileum in the case of avians, that's the site of in the small intestine where the amino acids and peptides, dipeptide and tripeptides are absorbed. It's very efficient. And however, some of them will not be absorbed because probably they are not yet fully digested, or maybe they did not have a chance to be absorbed by the transporters. They will go to the large intestine. The large intestine, undigested proteins can be utilized or fermented by microorganisms, and they secrete the proteases, and the protein becomes small peptides or amino acid. So there will be some free amino acids and small peptides in the lumen of the colon. So the question is, what happens to that, to those in the colon? Okay. How I learned, okay, I learned as a student in 1990s, I learned that in the colon, there's no amino acid absorption. There's no peptide absorption. Of course, at that time, we did not know that dipeptide and tripeptides can be absorbed in the small intestine. We just thought amino acid is the only end product that can be absorbed through the small intestine. But anyway, how about in the colon? More recent research shows that actually the epithelial cells lining the colon, okay, they expressed the similar amino acid transporters. Okay? So does that mean that we can see the amino acid absorption in the colon? Hmm, interesting. There are not much research going on, but you can still see some number of papers showing this, that the colon will have amino acid transporters. Okay, here I actually uploaded these two papers for you to read in case that you're interested. But you can see that amino acid transporters are expressed in the colon epithelial cells here. In the case of review paper or this research paper. And there, if I read those papers, the message here is those amino acid transporters found in the colon epithelial cells, they have high capability. However, they have a low affinity. So they're capable of transporting amino acid, but their efficiency is poor. Okay? So I cannot say there is no amino acid transport absorption in the colon. I should say there may be some amino acids can be absorbed in the colon. Okay? But if you talk about the significance, the amount, is that biologically significant or nutritionally significant? That's the question. Okay, but I still cannot say, oh, there's no amino acid absorption in the colon. Okay, so if you're interested in this area, I think there will be a lot more things to learn. You may explore your research career in this area too. Okay. Okay, so we talked about okay, what happens to those amino acids and small peptides once they are digested. I mean, they are obtained from the protein digestion. So they will be efficiently absorbed through the epithelial cells on the small intestine. Okay? That's what we learned. However, here, one interesting thing, not all the amino acid that were absorbed by the enterocyte will go to the systemic circulation. So that means some portion or a good portion of those amino acids from the protein digestion will disappear in the intestine or near the intestine. In other words, some amino acids or small peptides will be used by the intestine or somewhere in the close to the intestine. So this could include not just the intestine, but stomach or pancreas or spleen. Okay? And so those tissues right before the amino acid reach the liver, okay, they some amino acid could be used by these tissues. Okay? So we call that, collectively call that first pass metabolism of nutrients. So some nutrient, once they are absorbed, they will be just used by or disappeared in the intestine or pancreas and spleen, even before they reach to the systemic circulation. Okay? Well, it is reasonable to say the intestine, that's also, it has a lot of living cells and immune cells as well. They need to use nutrients. So, they need to use amino acid to make proteins, important enzymes, and so on. So there will be the first pass metabolism of amino acids. Okay? But how much? So it is interesting. In animal nutrition, probably in some sense in human nutrition as well. In animal nutrition, we have something called amino acid requirements. like a lysine requirement or methionine requirement. And we know, I think we want to say we know that we know the ideal amino acid ratio that we want to have in the diet so that animals can use those to build the protein most efficiently. And we call it ideal protein. Well, it's based on feeding studies or empirical studies. So I should question, do we really know what amino acid profile we should have in the diet so that animals can use them to build the proteins for in the muscle to grow well? Okay, I'm just challenging to say this because earlier we did not have enough information or knowledge about how much amino acid will be disappeared or catabolized by the intestine even before they enter the blood circulation so the muscle can use them for building the muscle tissue. So this knowledge about first pass metabolism is actually very important. But also, it's challenging the traditional nutrition knowledge that we have related to the requirement or ideal amino acid profile for the body weight gain or lean gain, muscle gain. But there are actually it's not that easy to do research to find out how much amino acid are catabolized by specific tissue like intestine. But there are very dedicated teams, research teams in the US and then also in other countries contributing the very important research data to us. Okay? So we know, okay, even though these values need to be updated and always validated, but we have information about first pass metabolism of amino acids. But here, I put one question here. What about gut microbiota? This is another important challenge here we have. Okay, we cannot say that there's a first pass metabolism of amino acid. Those amino acid disappeared after being absorbed, after being digested, and before entering the blood circulation. All those disappeared amino acids are used by intestine. We cannot say that because there's another user that we have been ignoring. But we know a lot more about them now in this state during the last 5 years. Okay? But those are the bacteria, especially those bacteria associated to the mucosa or colonizing the intestinal linings. So we call them mucosal bacteria or mucosal microbiota. They will utilize those free amino acids and they will use small peptides even before they are being absorbed. Okay? So once protein is digested, becomes amino acids, before they being absorbed into the intestinal epithelium, the mucosal microbiota, bacteria, will use them. So there's another user. But do we know how much amino acids can be used by mucosal microbiota? Do we know? Oh, well, that depends on what kind of bacteria are dominating there too. It's a very complicated thing, but this is something that we should not ignore. And this is also the area of research we should learn more about. Okay? This is a rather old paper from Dr. Peter Reeds and Doug Burin. They are very dedicated research team. Dr. Peter Reeds is not active anymore, but Dr. Doug Burin is still active, working at um, Baylor School of Medicine in Houston. They are one of the leading research team measuring this. Okay? In 2000, they put this paper and with the tables showing intestinal first pass utilization of dietary amino acid. What percent of intake will be used by the intestine? Okay. In the case of human, in the case of pigs, the human is splanchnic metabolism, including intestine, spleen, and the kidney. In the case of no, repeat. In the case of human, intestine, spleen, and pancreas. Okay, not kidney, right? In the case of pigs, only intestine. Okay. If you look at the lysine, okay, if human or pigs ingest 100 grams of lysine, 30 grams, 30% will be used by intestine or by the splanchnic bed, including intestine, pancreas, and spleen. Well, that's a significant amount, right? So we should not ignore this first pass metabolism. Wow, look at the glutamine and glutamate. In the case of pigs, almost all of them actually will be gone in the intestine, and they don't actually reach to the systemic circulation at all, 96%. So the first pass metabolism is very significant. We really need to understand about this if we want to modify the feed and make the animals grow faster, helping the lean gain, muscle gain, we need to understand what portion of those dietary amino acid will be used by intestine. When you say intestine, you're in 2000, we thought it's just intestine, but now good portion of first pass metabolism will be done by microbiota or mucosal bacteria. Okay? So there are another users here. This is a newer paper by Dr. Guoya Wu, retired from Texas A&M, okay, in 2021. So he collected information also from his own research, and he updated this table. Updated table by Doug Burin and Peter Reeds, but very similar. In the case of glutamate and glutamine, 96% are metabolized and during the first pass metabolism, including intestine and mucosal microbiota, even though we don't know the split. You can see the other values. Lysine, about 30%, 20 to 30% are gone. In the case of human, more than 30% to 40% are gone in the case of the pigs. Right? So significant amount of dietary amino acid are used in this first pass metabolism, including intestine and also microbiota. Okay, so this is very short, but I wanted to encourage and challenge you that amino acid from protein digestion, the animals, we or human, we cannot use all of them because even before we have access to those amino acid, actually intestine or the bacteria, even before intestine, you will use significant portion of that. Okay? So that's interesting challenge, and that's the area of the research is still lacking and can be very interesting area for you get involved. Okay? I'll stop sharing the screen here. Okay, I'll stop here, have a break, and then we'll come back. We'll talk about how we measure the digestibility of protein. Okay, there are different research methods to do that, and that's interesting to know. Okay, and it could be a lot related to your own research as well. Okay, thank you, and see you soon.